Hi, I'm Jonathan Teague, and I'm going to talk to you a bit about my PhD research at the University of Bristol. My project is on hyperspectral imaging as a tool for assessing coral health utilizing reflectance and natural fluorescence. So just to quickly start, a quick recap for those of you who know about coral, and maybe this is an introduction to those of you who don't. Coral is an animal that has a symbiotic relationship with an algae, colloquially known as Symbiodiniaceae or Zooxanthellae. And the relationship is briefly outlined in this image, in that coral gives shelter and waste products that the zooxanthellae can use to undergo photosynthesis and give the energy to the coral. So it's very much an exchange relationship. Corals also contain fluorescent proteins. The reason for this is not universally accepted, but here are a couple of theories. So one is to attract prey, one is converting high energy photons of light and re-emitting them at lower energy for photosynthetic use. So taking a wavelength that's not currently being able to be used by photosynthesis and converting it to one that can. And the last one is acting as a sunscreen. So taking in harmful UV radiation, reflecting it away or absorbing it. So bleaching. Bleaching more sort of generally refers to the expulsion or the removal of the algal cells, so the Symbiodiniaceae or Zooxanthellae, from the coral, so it expels them. And crucially, there's also a loss of color because the Symbiodiniaceae accounts for most of the coral's color. Without this, coral tissue is transparent. So as coral bleaches, it gets rid of these algae, and what you're seeing is the tissue, which is transparent, and then underneath that is the calcium carbonate skeleton, which is obviously white. So that's when corals bleach, that's why they turn white. And so looking at this color change, you can use that as a metric to understand how bleached corals are. And just as a quick sort of reference, if you can see a bleached coral by eye, it's lost about 70% of its zooxanthellae. And so if you do this, you can start to look at, well, how can we detect color and how can we look at color better? And so there are a couple of different, these are sort of predominantly, these are the surveys currently in use that look at color or spectral imaging. So anything to do with light and imagery. So you've got things like satellite images, which use hyperspectral and multispectral imaging. You've got aerial surveys, same thing. They can do those above the water. Obviously, those two techniques require some corrections in order to account for the atmosphere and the attenuation of light through water. And then under the water, you've got things like UUVs, so ROVs, AUVs, any kind of autonomous vehicles. You can do spectroscopy, you can look at pictures. And so sort of where this slots in is an accompanying tool to all of those things. So hyperspectral, I'm just going to briefly outline what hyperspectral imaging actually is. So a hyperspectral camera is essentially, it's a camera that consists of two parts, a camera slash imager and a dispersive element. So that's something that can split light into its incoming wave, into its wavelengths and bands. So that can be a prism, diffraction grating, or a filter. Essentially what this camera does is, rather than your typical camera that you have on your phone or GoPro or whatever, takes a picture in 3 channels, red, green, blue. This camera takes that to the extreme and does hundreds of those bands. So it's not just red, green, blue, it's all the way through the spectrum essentially. And what you're doing then is, if you have your imager and you take a picture, the X and Y refer to your actual your image, and then lambda, which is this Z in this plot, is your wavelength. So what you're doing is you're taking pictures of the same thing through the different wavelengths. And this is just an outline of just some of the different techniques that you can use to look at hyperspectral imaging. What we're going to sort of focus on is tunable filters and wavelength scanning. And so I'll go into that now. Just as a quick thing, hyperspectral images, obviously due to their complex nature and the mechanics involved in it, are generally not cheap instruments. They're fairly expensive. So they can range from about £20,000 to up to £120,000. And so that presents a problem for marine scientists and people wanting to use them, in that you've got a very expensive tool that you don't necessarily want to put underwater, because if it leaks, or the casing for it leaks or anything like that, you've suddenly got a very large financial sort of risk involved. So part of the project was to look at building cheaper sensors that could be used underwater. So one of the things we did was took a Nikon DSLR, so just a normal Nikon DSLR, which has the RGB channels. And what we do is we disassemble it and we take the CCD out. So the CCD is the thing in the camera that actually takes the picture and takes the incoming light and turns it into an image. And what you want to do is, what we did is we removed the bi-air filter. So the bi-air filter is the thing that makes it an RGB camera. So it's a matrix of essentially filters that filter out light into the 3 channels, red, green, blue. And so what you do is you remove that, and then underneath you basically have a monochrome CCD. So this is, rather than having the 3 channels, it has no channels and it just takes in all incoming light. And then you reassemble it, put that back together, and then you've got a monochrome camera. And then what we're doing is you're putting a filter that goes from 450 nanometers to 650 nanometers in 2 nanometer bands. So essentially what that means is it's sort of like a rainbow. It's a gradient of color across the sensor. And what that means is you can then use that for hyperspectral imaging. So the image in the middle is a picture of a fluorescent light. So what you can see here is you can clearly see some of the bands here, which correspond to the graph on the right. So what you can see here is that these are the fine bands that you can see, and the peaks relate to the brighter areas. And so how do you take that sort of imaging and that technique, and how do you actually make an image? So one of the things you do is you set your scene, and what you want to do is you want to image right across your scene so that you have every part of your scene or object, so the thing in the middle, at every point along the filter. So this is sort of what that looks like. So you've just got some Lego things in the middle there. What you do is scan all the way across. And then what we do is we take all of those images. So we're taking pictures all the way along, and we put those into photogrammetry software. So the photogrammetry software, what that does is it stitches all the images together. And then based on, we know where each image was taken relative to the space, we have camera positions. And so what we can do is we know that on camera position 1, the top left pixel, for instance, is in the red, and the top right pixel is in the blue. And we can basically map the pixels and where they are on the scene to be able to get spectral data from that. And so what that looks like in terms of an animation is, here we go. So in the bottom right, you've got wavelengths, and that's denoted by the number that's 400, 500 now. So that's in nanometers. So as you can see, it's kind of scanning across, but crucially, the scene in the middle, the objects in the middle, are imaged at every single part along that filter, which gives us spectral information. And so what that means for in the marine environment, for instance, one of the sort of most comparable techniques is looking at spectroscopy, or using a spectrometer underwater. And what that might involve is swimming down, imaging a coral a couple of times, moving on to the next coral, image that a couple of times, the next one, image that a couple of times. And then what you're doing is you're using, say you've done 3 measurements per coral, you're averaging those 3 measurements to get a general idea of the spectral profile of the whole coral, which is fine. But say if it's a very large coral and you've only got 3 data points, it starts to get a little bit, the data is not as accurate as it could be. And also it's very time consuming to do this. Whereas with a hyperspectral imager, you could just swim over the reef. So you do it in a raster pattern, very similar to what you would do if you were doing a photogrammetry survey. And you would just swim across the top of the coral, and rather than having 3 points per coral, you would have thousands of spectral points, because it's based on pixels. So you've got thousands of pixels in your camera every time you take a picture. And so when you're looking at a coral, rather than having 3 points, you could have hundreds of thousands of points. And what that means is you're starting to get a better idea of the whole spectral profile of a coral, rather than just 3 points. And so hyperspectral imaging is currently used in marine applications. So you have, obviously, as I said before, the satellite data. So that gives you huge areas, but at low spectral resolution. And then you've got the airborne stuff, so in planes and things. So you start to look at small areas, but you get that higher spectral resolution. But the problem with these 2 techniques is that they rely on corrections. So because you're imaging either in space or in the atmosphere somewhere in a plane, you have to account for how the light is attenuated by first the atmosphere, so it could be scattered off particles, and then the water itself. And so one of the things you do also need to do is ground truthing. So you would take measurements underwater with a spectrometer or a hyperspectral camera to basically verify that your algorithm that you made to correct for your data is representative of the spectral profiles underwater. And so when you're doing it underwater, you have the smallest areas, but you have the highest spectral resolution. And also you can look at other things like fluorescence, which you wouldn't be able to see from space or in a plane, because you need some sort of excitation light in order to elicit the fluorescent very brightly. And so what we did is we looked at bleaching experiments in the lab. So what we've done is we've taken some corals that we've got from London Aquarium and split them into 3 groups. So we've got a pre-treatment group, a control group, and a bleach group. So the pre-treatment group, so we've taken the cells for these. So this data also takes into account cell data. So pre-treatment, the cells were harvested immediately, and then obviously bleach and the control in separate tanks just went for 3 weeks. And the bleaching tank, we turned the temperature of the water up by 2 degrees each week in order to basically force artificial thermically induced bleaching. And so this is a sort of look at the data. So on the left here, you've got fluorescence. So you can see in the pre-treatment and control, you've got the 3 peaks. So you've got peak 1, which is the excitation source. So this is the UV light that we use. So that's at 445. And then you've got the second peak here, which is your green fluorescent proteins. And then you've got a third peak there, which is chlorophyll fluorescence. And then on the right there, you've got a reflectance. So again, we're using an artificial white light, and the white light contained some of the blue light that we used in the other one. So you can see that same peak there, number 1. Number 2, you've got sort of 2 peaks there, which are representative of the same pigment. And then there you have your, that is chlorophyll absorption. And then there's another pigment here at 7, just after 700. And so this is what that looks like in terms of the fluorescence before and after. So just to get an idea that you can see that there's clearly on the green fluorescent protein at about 527, just drops away completely, and the chlorophyll fluorescence at 685 reduces drastically also. So you can see that these signals do give you an idea of sort of bleaching. So this is the cell counts that we did in the experiment. So you've got the red is the bleach, green is the control, and pre-treatment is blue. Now you can see there's a difference in that ideally the control and pre-treatment would be the same, but obviously some bleaching has occurred in the control tank as well, which is unfortunate, but it's still significantly higher than that of the bleach group. So it's still representative. So what that looks like if you plot the final cell count versus the final intensity of each of the groups, and you average it. So you can see there's 3 very distinct groups there in that it seems to be a proportional between cell counts and intensity. So the more cells there are, the higher the intensity, and that makes sense. And so what happens then if you try to map through time? So you're imaging the same coral with the pigment through time. And so this is what that starts to look like. So we've got 676 is chlorophyll absorption and reflectance, and 760 is pigment also in reflectance. And so you can see there's not that much of a significant difference between the two. You can see there's some fluctuations that are sort of mirrored throughout both. But if you start to look at the fluorescence data, you start to get a slightly different picture in that 511 is your green fluorescent protein and 685 is chlorophyll. On the bleach sample for green fluorescent protein, you can see it's got a very sharp drop. And when compared to the control, there is still a drop, but it's nowhere near as drastic as the other one. And this sort of alludes to the fact that green fluorescent protein is probably one of the first things to be expelled during the bleaching process, and so makes a very good marker for how bleached coral is. So what you want to be doing is you want to be imaging the same coral time and time again. So if you're doing this, as this moves on to sort of conclusion, in that they obviously play a crucial role in coral, and actually understanding how they work and their importance, will help us determine how good of a marker they can be. And also that hyperspectral imaging does allow for fluctuations in coral pigment to be monitored and recorded, obviously. So if you were to do this, moving on to future work, something that we want to do is to image live reefs and imaging that same reef time and time again. So ideally you'd want to do it, at least for, once a month for a year, in order to account for seasonal fluctuations in pigments, and then keep going back to that same reef to get a baseline, and then collecting your seasonal data to be able to see the changes in the coral color. And obviously this leads into we would want to be doing more of the lab experiments to sort of back up and look at these different corals. And obviously there's a picture of the system there it's ready to go. Obviously once lockdown is lifted, we can go and do field work. And so there's some references there. Thank you for listening. If you have any questions or anything like that, feel free to contact me. I've left some details there on the screen. Thank you very much for listening.